Mendel learned a lot from his monohybrid crosses, or crosses that followed only one trait. But his next step was going to be crossing organisms that differed by two traits. And the question he really wanted to understand was whether traits were inherited together. So if they occurred in a combination in the parents, did they always have to be inherited in that same combination in the offspring? For example, if the parents were purple flowered with tall height, and the other parent was white flowered and short height, then could all of the future offspring only have purple with tall and white with short? So that was the linked hypothesis, or possible pattern of inheritance. So let me write that there. So were traits linked, or were traits independent? If the traits were independent, that would mean that purple and tall was not always inherited together. So you could have offspring that were purple with short, or white and tall, or you could also have the original combination. So purple, tall, white, and short. So even though the parents had purple with tall, you could get any possible combination in the offspring because the traits are inherited independently. So that's the two possibilities he wanted to test. I've got this one underlined because what Mendel's going to find is that the traits are independent, at least the traits that he studied. So to do this, he did, again, a cross of two traits, and that's called a dihybrid cross, di for two. So they're going to differ for two traits. So in this example, he's following seed color and seed shape. So two different traits. Seeds are either yellow or green, and green is recessive, or they're round and wrinkled is recessive. So those are the possible alleles, yellow or green allele, or the round and wrinkled allele. So he starts with true breeders. So true breeding for two traits. So true breeding yellow round, and he crosses that with a true breeding green wrinkled. On this slide here, we have two different Punnett squares. This is the Punnett square if the traits are linked. This is what the result would look like. This is the Punnett square if they are independent. So we're starting with the same thing here. So again, the true breeding yellow round with the true breeding green wrinkled. So because they're true breeding, remember that means they're homozygous. So yellow round is homozygous yellow, homozygous round. Notice we're having to follow 4 alleles now because it's a dihybrid cross. The other parent is true breeding green wrinkled, so they are homozygous recessive. And the same thing over here, because remember this is the same exact cross, just the outcome's going to be different because if it's linked, it's going to be a different outcome than if it's independent. So at first you can't tell. So in the P generation, we cross-fertilize those or hybridize them, and then we look at the F1 generation. And at first there's no difference. So you're going to see the exact same result. They're all yellow round. And that's because this one plant, in its gamete, it could only have a big Y and a big R, and this one could only have a little y and a little r. And again, that's same on this side. And so you get the heterozygote. So all yellow round. The measurable difference between linked and independent happens in the F2 generation. So the linked, basically, it gets that name linked because it's saying that there is a physical connection between the yellow allele and the round allele, and a physical connection between the green and the wrinkled allele. So that means that even though this individual has these four different alleles, these two alleles always have to go together, and these two alleles always have to go together because they're physically linked. So when they go to make gametes, they only make either the big Y with the big R or the little y with the little r, and the other parent's the same, self-fertilization. So if they're physically linked like that, because that's how they inherit it from their parents, then you would only see a ratio of 3:1 yellow round to green wrinkled. And the key here is that What did the parents have? Yellow round. What did the grandparents have? Yellow round. Green wrinkled. That's the combination we saw in the grandparents. So these are the same combinations of traits that were present in previous generations. And again, that's what's going to happen if the traits are linked. The other possibility is that the traits are independent. That means that there is no physical connection between the Y, the big Y, and the big R. So that means you can have the big Y with the big R, but you can also have the big Y with the little r. You can have the little y with the little r, but you can also have the little y with the big R because there's no physical connection between the alleles. They're totally independent. So if you look across here, this is showing all the possible gametes, big Y with big R, big Y with little r, little y with big R, little y with little r. And again, down the side, the parents are the same, so it's the same. And then just like with the Punnett squares before, we combine them in the middle, and that gives us our traits. And if you go through and count these, you'd find that there's a ratio of 9 yellow round to 3 yellow wrinkled to 3 green round, and then the last one here kind of is cut off a little bit, a ratio of 1 green wrinkled. So a 9 to 3 to 3 to 1 ratio. And so notice that we have yellow round and green wrinkled, which we saw in previous generations. These are called the parental phenotypes. So yellow round and green wrinkled we saw in the parent and the grandparents. But, and here's the big thing, we see new combinations of traits. Yellow with wrinkled was not a trait that we saw in previous generations. Neither was green round. So these new combinations of traits are appearing in the offspring. That's if the traits are independent. When Mendel did the cross, okay, so these were the two possibilities or predictions of what would happen. This is what's predicted to happen if it's linked. This is what's predicted to happen if it's independent. When he looked at the data, and here's the Mendel's data down here, he found 315 yellow round seeds, 101 yellow wrinkled, 108 green round, and 32 green wrinkled. And this is about a 9 to 3 to 3 to 1 ratio. And again, the big thing here is that we see the new combinations of traits, yellow with wrinkled, green with round. So his conclusion was that the traits were independently inherited. And this is what's called Mendel's law of independent assortment. Alleles of different genes, so seed color and seed shape, assort independently from each other during gamete formation. So that allows the big Y to go with the little r, or the little y to go with the big R. They get new combinations during gamete formation. So let's walk through a dihybrid cross. So this is a question. Pea plants are heterozygous for flower position and stem length, and they're allowed to self-pollinate. 400 of the resulting seeds are planted. How many offspring would you predict to be dwarf with terminal flowers? So my parents, so start with my parents, are double heterozygous for these traits. So now I need to decide which gametes they're going to make. So every gamete should have one allele for each gene. So let me write that here. This is a place where some people get confused. So in other words, they should have one A and one T. So one allele for each gene. So the possible gametes are this big A with this big T. That's one possible gamete. This big A with this little t is another possible gamete. The little a with the big T is another possible gamete. And lastly, the little a with the little t is another possible gamete. Now, because this is a self-fertilization, the parents are going to be the same. They're going to make the same gametes. So for a square like this, you have to, or for a dihybrid cross rather, you have to draw a much bigger Punnett square because you have 4 possible gametes for each parent. So your Punnett square should have 16 total boxes on the inside. So now again, because these parents are the same, I'm just drawing the other parent's gametes down the side, and then I combine. So combining this gamete with this gamete, and again, just doing the same thing. Okay, almost there now. Okay, so now I've filled in my Punnett square. Now I need to interpret what happened. So I'm going to start by looking for axial tall. So this one would be axial tall, this would be axial tall, because remember, those are the dominant traits. So I'm just looking for dominant alleles there. This one would be axial tall, this one would be axial tall, this one, this one, this one, this one, and this one would be axial tall. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9 axial tall. Okay. Let's see. Next one, axial dwarf. So now I'm looking for one dominant trait and one recessive. So there's the dominant axial with recessive. Oops, there's another one. And there's another one. So there's 3 axial dwarf. Now terminal tall. So now this one's recessive and this one's the dominant trait. So looking for those. So this one's terminal tall, terminal tall, terminal tall. So there's 3 of those. And then last one is both recessive. So terminal dwarfs. Change fence here, and there's only 1 of those. So let me rewrite that for you because that was hard to read. The terminal dwarf, there's only 1. So notice that's a 9:3 to 3:1 ratio. That's what you would expect for a dihybrid cross, which this happens to be. And so the next part of the question is, how many of 400 seeds would you expect or predict to be a dwarf with terminal flowers? So this is now converting the ratio into an actual number, and depending on your math background, you may or may not already know how to do this, but I'm going to walk you through this. So 9:3 to 3:1, remember, is also expressible as a fraction. So there's a total of 16, because 9 plus 3 plus 3 plus 1 is 16. So that means that I could also say that 9/16 of them will be axial tall, 3/16 of them will be axial dwarf, 3/16 will be terminal tall, and 1/16 will be terminal dwarf. So that's converting the ratio to a fraction. Okay, so again, 9 plus 3 plus 3 plus 1 is 16. So that's what makes the bottom of my fraction be 16. And then this number is the top of the fraction. Now that I know that 1/16 of them should be terminal dwarf, then I need to find out what 1/16 of 400 is. And that's just a little bit of basic math. 1/16 times 400, and that is equal to 25. So 1/16 of 400 is 25. Therefore, I would predict that I would see about 25 terminal dwarf offspring from 400 total offspring. You're going to need to be able to do this math in the lab, so make sure you really pay attention to that math there.